video we will be going to study about the formation of the hemoglobin, because hemoglobin, it is an important part of our cardiovascular system, because it's present in hemoglobin, it's present in the red blood cells, RBCs. Okay, so it has an important function, because one of the important functions of the hemoglobin is transport of oxygen and carbon dioxide. So it transports oxygen and carbon dioxide, oxygen from lungs to the tissues and carbon dioxide from tissues to the lungs, and then it's expired from the lungs. Okay, so it has these two are the important functions. I will explain how it functions. For example, this is the alveoli of the lungs, and this is the blood vessel, and this is the cell where it has to reach. Okay, so we inhale oxygen by inspiration. So this oxygen here, it has the less partial pressure, and here the oxygen has the, this partial pressure of oxygen here is high PO2, and here there is low PO2, and this is the blood, and in blood there is presence of the RBC. So this is the RBC, and RBC contains hemoglobin, Hb. Okay, so the oxygen, when we inhale the oxygen, this oxygen will move from the alveoli of the lungs to the bloodstream because of the difference in the partial pressures, and here from the blood this oxygen will bind to the hemoglobin, and this hemoglobin will carry oxygen to the cells. Again, here PO2 is high, here PO2 is low, so it will move into the cell. So here there will be presence of the oxygen. Now second step here is digestion. So during digestion, at the end there is a formation of like glucose. So glucose will bind with this oxygen, and it will form ATP plus water plus carbon dioxide. Okay, now this carbon dioxide, this has to move out of the cell, because carbon dioxide is not that important in our body. Its high concentration is not important for us, it's not beneficial for us. So this carbon dioxide, this has to be moved out of our body. So the RBC here it will bind to the carbon dioxide, and then it will go here, and again here the partial pressure of carbon dioxide is high, here partial pressure of carbon dioxide is low, so carbon dioxide will move out of the lungs by expiration. So this was inspiration, this is expiration. So this is the function of the hemoglobin, how the hemoglobin functions in our body. So now we will go to the structure of the hemoglobin. So basically the hemoglobin, it is heme plus globin. So globin is a protein part, and heme means iron, you can say Fe, and protein. So the protein part, it contains 2 alpha chains and 2 beta chains. Okay, and there are 4 iron molecules which are present in one molecule of hemoglobin. So if I will draw the structure of the hemoglobin like this, for example this is the alpha chain, and this is the second alpha chain, this is the beta chain, and this is the second beta chain. Okay, so this is beta chain, this is also beta chain, this one is alpha chain, this one is alpha chain. So this part is the protein part of the hemoglobin, and it also contains iron. So this is the iron, or we can say it is the heme part. So in hemoglobin there is presence of heme part, which consists of iron, and globin part, which consists of proteins, 2 alpha chains and 2 beta chains. That is the molecular structure of hemoglobin. Okay, so if we will say how oxygen is carried, I already told you Hb, it will bind with oxygen, 4 oxygen molecules, and it will form HBO8, which is called as oxyhemoglobin. So this is oxyhemoglobin. So one molecule of hemoglobin will carry 4 molecules of oxygen. Okay, so that's oxyhemoglobin, and this is a reversible reaction. Okay, so that means 8 oxygen atoms can be carried by each hemoglobin molecule. 8 oxygen atoms carried by each hemoglobin molecule. Okay, now this hemoglobin can bind with carbon dioxide also, Hb plus CO2, HbCO2, which is called as carbohemoglobin. So when it reacts with carbon dioxide, it forms carbohemoglobin. One more thing is it can react with carbon monoxide also. Okay, so Hb plus CO, it forms HbCO, which is carboxyhemoglobin. Carboxyhemoglobin. So carboxyhemoglobin is a stable compound. It is stable compound. Okay, so carboxyhemoglobin, it can't carry oxygen and carbon dioxide. Okay, it can't carry oxygen and carbon dioxide. That's why it causes anosmia, and which further causes suffocation. Because because oxygen can't bind, further it will cause the suffocation. And if carbon monoxide and oxygen both are present in the blood, this hemoglobin has high affinity for the carbon monoxide. That's why the carbon monoxide is dangerous for us, because it can cause suffocation. Okay, and carboxyhemoglobin is formed generally, the carbon monoxide which is formed by the incomplete combustion. For example, ethane, C2H6 plus oxygen, they combine together, they form CO plus 3 times water plus carbon. So here there is the formation of the carbon monoxide, and this is a incomplete combustion. Okay, so this is fuel plus oxygen, they form carbon monoxide plus water plus carbon. Okay, so that is how the carboxyhemoglobin, then this carbon monoxide, it reacts with the hemoglobin which is present in our blood. If there is ingestion, accidental or any other ingestion of the carbon monoxide, so it will form carboxyhemoglobin, which causes suffocation, which is very dangerous for our health. Now our topic is formation of the hemoglobin, how hemoglobin is formed in our body. So basically it is present in the RBCs, so it will be formed in the RBCs. Okay, so it is a iron containing, or we can say oxygen transport metalloprotein in the red blood cells, or erythrocytes we call them. Okay, and it is present in almost all the vertebrates. Okay, and some in the invertebrates also. It carries oxygen from lungs to the rest of the body, that I already discussed. Okay, so it develops in the cells in bone marrow cells. So it develops there. Okay, so hemoglobin, it develops in the cells in the bone marrow that become red blood cells. Okay, and when red blood cells die, hemoglobin, it is broken up after the death of RBCs. Then this Hb breaks down. It's formed in the red bone marrow cells, and after death of RBCs, because RBCs have a lifespan of 120 days, after death of the RBCs, this hemoglobin, it breaks down. Okay, and iron is removed and transported to the bone marrow, because it contains iron and protein part, and iron is removed and it again goes to the bone marrow. Okay, iron goes to the bone marrow, and by the proteins which are called as transferrins. So transferrins transport iron from the dead RBCs to the bone marrow. Okay, and they again use it in the production of red bone cells, and protein part, it's converted into biliverdin, which goes into the liver. Okay, so protein part, it's converted into biliverdin, which is transported to the liver, and then through the liver it gets excreted through feces. Okay, that is the destruction of the hemoglobin. Okay, now there are various risks of high and low hemoglobin levels in our body. Like if there is low Hb level. Okay, so low Hb levels, these are generally associated with kidney disease or liver disease or anemia. Anemia has different causes. Okay, and elevated levels are, we can say, as high levels of Hb, and these are associated with chronic lung disease or dehydration or heart failure. Okay, so these are the conditions where there is low hemoglobin and where there is high hemoglobin. Okay, now I already told you hemoglobin, it's made up of 4 polypeptide chains. What are those chains? 2 alpha chains, 2 beta chains, and each chain is attached to the heme group. Okay, which is composed of porphyrin, and porphyrin, it is an what we can say, an organic ring like compound which is attached to the iron atom. Okay, and these iron porphyrin complexes, these coordinate oxygen molecules reversibly, and ability to directly related to the role of hemoglobin oxygen transport in the blood. Okay, so hemoglobin, it is synthesized in a complex series of events. It is a complex process, complex series of events are there. Okay, so that I will explain here. So we are going to study about the formation of the hemoglobin. Now, for example, this is an RBC. This is a red blood cell. Okay, first thing is there is a transferrin receptor. I told you the by transferrin receptor this iron part is transported. So here there will be presence of transferrin receptor. So this is an RBC. So here there is presence of the transferrin receptor. For example, this here is a transferrin receptor. Through transferrin receptor, what will come? Iron, Fe. Okay, and one more structure which is present in the RBC, that is mitochondria. So this is the mitochondria. Okay, so this iron, Fe, this iron will enter into the mitochondria. It's called as heme. Okay, and another source is ferritin. Ferritin is the another source of iron. Either it can be transported to the transferrin receptor, or it can come from the ferritin. Okay, so iron can come from transferrin receptor, and it can come from the ferritin also. So this is the iron that's come to the heme. Okay, now second step from here is glycine, which is present in the mitochondria. It reacts with succinyl coenzyme A. Okay, so glycine plus succinyl coenzyme A in presence of vitamin B6. Okay, in presence of vitamin B6, and there are series of reactions. Like first there will be formation of alpha amino levulinic acid, levulinic acid. Then there will be porphobilinogen, and this all occurs inside this cell. So I don't have space here, that's why I am writing it down. Okay, this porphobilinogen, it's converted into uroporphyrinogen. Then this uroporphyrinogen, it's converted into proporphyrinogen, and then this is converted into protoporphyrin, and then this protoporphyrin reacts with the heme, and it causes the formation of the hemoglobin. So I will again explain these steps. So this is the RBC, and this is the mitochondria of the RBC. So this is a transferrin receptor. Through transferrin receptor, iron will be transported, or it can come from the ferritin. Then it enters into the mitochondria. It remains here as a heme. And glycine plus succinyl coenzyme A in presence of vitamin B6, after a series of reactions, they form protoporphyrin, which reacts with the heme and forms hemoglobin. So this is one way by which the hemoglobin is formed in our body. Second is amino acids. Amino acids which are present in our body, these are carried to the ribosomes, and there they form alpha and beta chains. Means 2 alpha and 2 beta chains. Okay, means globin part. Okay, and this is one more method by which hemoglobin is formed. It reacts with the iron, then this globin part, and forms the hemoglobin. So that is how the hemoglobin is formed in our body. So this is the formation of hemoglobin in our body. This whole step, this is the formation of the hemoglobin. Okay, so how the hemoglobin is formed in our body? So first there will be a transferrin receptor which transports the iron from outside of the cell to the inside of the RBC. Okay, and there is one more source of the iron, which is ferritin. Okay, ferritin is the other source of the iron, and this iron enters into the mitochondria of the RBC. This is the mitochondria of the RBC. Okay, this is the mitochondria of the RBC, and in mitochondria there is presence of glycine plus succinyl coenzyme A, which react together in presence of vitamin B6, and after a series of reactions they form protoporphyrin. This protoporphyrin then reacts with the heme. okay, which has already been transported from outside and forms hemoglobin. And another way by which hemoglobin is formed is from the amino acids which are transported to the ribosomes, where protein synthesis occurs, means 2 alpha and 2 beta chains are formed, globin part. This globin part then reacts with the heme part and gives rise to formation of the hemoglobin. So this is how the hemoglobin is formed. Or we can say the heme part is synthesized in a series of steps in mitochondria and cytosol, okay, because heme part it's synthesized in the both parts, both the organelles, mitochondria and cytosol, and this is formed in the immature blood, red blood cells, okay, and while as globin parts are synthesized by the ribosomes in the cytosol. So globin part in the ribosomes and heme part is synthesized in the mitochondria and cytosol. This is mitochondria and this is cytosol, okay. And production of HB, it continues in the cell throughout its early development, means from the pre-erythro or pro-erythroblast to reticulocyte, which we already stated in the hemopoiesis, okay. So production of the HB, it continues from pro-erythroblast to reticulocyte. It occurs in the bone marrow, okay. And it's a complicated process. This takes place in RBCs. Amino acids, these are assembled in the ribosomes, okay. Here we can see the amino acids which are assembled in the ribosomes, okay, and they give rise to 2 alpha and 2 beta chains, which is the globin part. And heme molecule, it's protoporphyrin, and it's formed by the combination of glycine and succinyl coenzyme A. You can see here glycine and succinyl coenzyme A, okay, by their combination in presence of the vitamin B6, okay. And transferrin, it gives iron which will combine with the heme molecule, okay. Transferrin, it gives iron which will combine with this heme molecule, okay. And heme molecule, it consists of protoporphyrin, I have written here protoporphyrin, protein and iron, and globin, it consists of 4 polypeptide chains, okay, alpha, beta, 2 alpha chains and 2 beta chains. So this is how the formation of hemoglobin occurs in our body.